Welcome students. Today we are going to learn Standard 9 Subject Science and Technology Part 2 Lesson number 17 Introduction to Biotechnology. So let's begin with the topic Biotechnology. With the help of biotechnology, it has become possible to produce plants and animals bearing some new characteristics in addition to their natural ones. The technique of bringing about improvements in living organisms by artificial genetic changes and by hybridization for the welfare of human beings are together called biotechnology. It includes the techniques of genetic engineering and tissue culture. Its uses are in the production of cash crops and improvements in varieties of cash crops, increase in the abilities of plants to withstand environmental stresses, vaccine production, early diagnosis of congenital diseases, organ transplant, cancer research, production of artificial skin, cartilage, etc. in the laboratories. Let us have glance upon what is mean by tissue culture. Tissue culture is ex vivo growth of cells or tissues in an aseptic and nutrient rich medium. Nowadays, a complete organism can be developed from a single cell or from tissue with the help of tissue culture technique. A liquid, solid, or gel like medium prepared from agar which supplies nutrients and energy necessary for tissue culture is used in this technique. Tissue culture technique involves the steps as shown in the figure. The first step is collection of tissues from the source plant. The parts like a bulb corm of a source plant can be used for starting the tissue culture. After that, it is followed by the primary treatment, then multiplication, shooting and rooting, that is formation of the shoot and root in the laboratory, then primary hardening means transferring the plantlets into the trays, and finally the secondary hardening where the plants are ready to be grown into the soil. So this is all about the tissue culture. Now we move forward with the topic changes in agricultural management due to biotechnology. Genetically modified crops, which are also called as GM crops, are being produced by introducing changes in the DNA of natural crops. Normally, such varieties are not found in nature, thus, new varieties are produced artificially. Different useful characters are introduced in such varieties. Also, by using biotechnology, different varieties which have the ability to withstand environmental stress can be produced. Some naturally occurring varieties cannot withstand environmental stresses like frequently changing temperature, wet and dry famines, changing climate, etc. However, GM crops can grow in any of such adverse conditions. The GM crops which are produced by the biotechnology are resistant to insect pests, pathogens, chemical weedicides, etc. And therefore the use of harmful chemicals like pesticides can be avoided into the field where GM crops are grown. Also, due to use of seeds of GM crops, there is improvement in nutritive value and decrease in loss of crops, which ultimately helpful in increasing the yield of crop. Due to such better quality seeds of GM crops, farmers all over the world are cultivating the GM crops on large scale, and the area under its cultivation is increasing day by day. The high yielding varieties of GM crops which are produced by biotechnology like banana, maize, rice, potato, soybean, tomato, cotton, apple, brinjal, papaya, beet, rose, tobacco, wheat, etc. are available. The examples of GM crops are as follows. The GM varieties for maize are MON 810, MON 863. For potato, it's Amphora. For rice, it's a golden rice. For soybean, it's a Vistive Gold. And for tomato, it's a Vaishali. And the well-known example is cotton, that is BT cotton. Along with the food crops, biotechnology is also useful in floriculture, nurseries, and forestry. Nurseries are necessary for various purposes like growing gardens on a large scale, afforestation, reforestation, etc. Seedlings have to be supplied in large numbers for all these purposes. It is profitable in such cases to produce plantlets with the help of tissue culture techniques. So look upon the applications of biotechnology in floriculture, nurseries and forestry. Tissue culture can be used to grow those plants on a large scale which bear flowers, fruits of excellent quality. Using biotechnology, fully grown plants can be produced in shorter duration. Plants can be grown on a large scale even if means of pollination or germinating seeds are not available naturally. For example, orchids or peach plants do not germinate, but these plants can easily be produced by means of tissue culture. The best benefit of using tissue culture for growing the plants is in a bioreactor, cells can be grown in a more nutritive medium and protected from pathogens. Also, large numbers of seedlings or plantlets can be produced in a short time using minimum resources and materials. The plants which are produced by tissue culture and genetic modification techniques are disease free. Plantlets produced by tissue culture of the meristem are virus free. The best advantage of using tissue culture is embryos formed by conventional hybridization technique between two or more varieties may not grow fully for some reasons. However, embryos produced by tissue culture technique always complete their growth. Using tissue culture technique, rare and endangered plants can be grown and thus can be protected from extinction. Thank you.